our next speaker, Shri Gautam Biswas, sir. Sorry, sir, for the delay.
Okay, it's okay.
Use of information and communication technology for teaching biology. Gautam Biswas, sir, has 25 years of experience teaching biology. Apart from teaching, he's in charge of Academy Advisory Committee, convener, Science Subject Committee, in charge of Green School Science Exhibitions, key resource persons for in-service courses and for PGT biotechnology teachers, KVS for capacity building workshops, coordinator in preparation of study materials in biology for 11th and 12th, question paper setter, moderator in various exams, an expert talks in various online programs, conferences such as World Earth Day, Women's Day, Mano Darpan, STEAM education, and application of technology in teaching learning. Is a key member of East India chapter. So we welcome you, sir, on this topic. And now it's over to you, sir. I'll make your...
Thank you, Suman ma'am.
Yeah, sir.
Thank you, Suman ma'am, for giving...
Sir, I request SP Singh, sir, to make me host so that...
Pardon, madam, बताएं क्या कह रहे हैं?
Sir, आप मुझे host बना दीजिए, please. You are the host, so I cannot share now.
अच्छा, मेरे को बताएं मुझे क्या करना पड़ेगा अभी?
Sir, Suman Joshi, the 3 dots were there, so please make me host.
3 dots.
Automatically, yeah. In participants list.
Yeah, मैंने आपका वो 3 dots आपके नाम के आगे था.
Yes, sir. And it's
waiting, allow to record computer. अच्छा, make host.
Yes, sir, please.
Right.
Yes.
तो ये कब हुआ? आप host तो आप ही हैं, madam.
नहीं, sir. I made you host, then only you could share.
Oh,
the
okay.
PPT.
I'm sorry.
Yes,
I'm
sir.
sorry. I was not
Sir.
And
Okay.
Gautam sir, मैं आपसे माफी चाहता हूँ कि आपका टाइम मैंने लिया.
Actually, that was really very enriching session,
आप
sir.
बोल रहे थे, हमें सुन रहे थे बहुत देर
बाद. You are the host.
It's okay, sir. It's okay. आपका पीपीटी देख के ऐसा लग रहा था कि और भी कुछ जानने को मिलेगा. It was quite good enough. And टाइम रहता तो और भी आप थोड़ा बहुत इंक्रीज कर सकते थे. बहुत दिन बाद मिला इस प्लेटफॉर्म में, सर.
Thank you.
Good health and mind.
Can I stay with the meeting and मैं आपके भी विचार जानना चाहता हूँ जो आप प्रेजेंट कर रहे हैं उसको मैं थोड़ा सा
Sure.
Thank you very much.
Please be with us.
सर मैंने आपकी आवाज सुन लिया सर. नमस्कार.
So
thank you, sir.
So Shubham ma'am, shall I start?
Yeah,
Can
sir.
I share my screen?
I have made you the host. Now you can share PPT, sir. Yeah.
Okay,
Thank you.
okay.
Thank you, sir.
So just before starting, I should thank Dr. A.K. Sharma sir for giving me opportunity. He has always been an inspiration for us from long back, since 2012, I feel so, when sir was in KV Gold Market as a principal. And really getting such a platform in order to share my views is obviously, I do feel myself lucky enough to be with you all. Well, as far as the topic is given to me on ICTs that can be used in biology. So I would like to share some of the important aspects. Just a minute. Yes, I think my screen is visible. Suman ma'am?
Yes, sir, it is visible.
Is my screen visible?
Yeah,
okay.
visible.
As the title of the webinar, Biology Unlocked, said by sir. So in this, I had just tried to select few of the ICTs that had been used or can be used out of many. There are many, and yet they are growing like mushrooms. We have to see which one fits best for our teaching. And hence I'm just going through few of them. Okay. So use of information communication technology for teaching biology. This a word cloud, and I feel many of you have used some of them or many of them. Am I right? From COVID and post-COVID, you can see we may have been using Jamboards or Mentimeters, Kahoot, and many such things. Now, there can be some new, there can be some old one. And what we do or how we can use this ICT actually totally depends upon the teacher and how he or she plans to teach in the class. So the next slide, I'm giving a general outline. This is not what you call a final rule that this is how you have to teach. You can make it in your own way. What we do, we just relate the topic in everyday's life. We may use some app. We may incorporate hands-on activities, if possible, either practical or we may use some apps over here, or create a multimedia material. We can use ICTs. Look for ways to bridge technologies and biology. So that time also we can use this, or utilize science games to teach biology. So wherever we find that there is a limitation in doing hands-on practical, we can shift towards ICT. Wherever we find difficulty in explaining a particular concept, we can use ICT. So ICT can be used. It can supplement teaching. It can help teaching. It can make the student understand it's best if you use it in a proper way. And let's see what are the different ways that how we can engage our students by these ICTs. Now, many of you have heard about Mentimeters. I don't know how many of you have used this Mentimeter. Now, you can either, if you are using laptop or desktop and having a spare mobile, you can scan the QR over here, or you can click into the link and you will get a question. Okay. So just I'm clicking it over here and we reach to a page. This Mentimeter is such a software or a tool or a platform where you can take some either polls or views, or it can be used for icebreaking a session. Whenever you start any online talks or webinars, in order to understand the feedback of the students, you can just use Mentimeter. It gives a real-time reflection. For example, for simplicity, I have asked over here, some part of the screen is forward. What do you understand by the term biodiversity? And there are few options. If you scan it in your with your mobile, the QR code in Google Chrome, you'll find that there is a Google Lens. In that you scan it, you will find the same thing. And if you get the same thing, you type some of your answer, what you feel like, anything, let it be wrong or right, does not matter. Just to show you a practical aspect that if you just enter certain terms and submit, what will happen? Can you try it? Is it possible? Let's see how interactive it is. Are you getting the scanner, or shall I go back to the scanning page?
Go back to the scanner.
Okay. Just a minute. Yes. Or you can also, without scanning also, you can go to Google and type menti.com. Okay, not Mentimeter, menti.com, and use the code 11229678. Are you getting this page? Very good. I'm getting some response, live form.
But
So
on the
see,
side, there is enter a word, enter another word, enter another word.
Yeah, you can word 3 words. You can enter 3 words.
But how to enter? It is not.
You have to enter the word and submit. You enter 3 words and submit at last. Say, so like that, if you just ask the student and the students give feedback, you can understand that how much they know or how much the depth of their knowledge, and you can introduce the topic, or you can just plan it that how you should proceed further. Now, after this, we can go to the responses. The responses we have seen already in the screen. That is in Mentimeter. Mentimeter is the platform in which you're preparing the question and sending it. You can put a poll or a word cloud or such type of feedback questions. You can just keep it and ask the students to answer while in the class, and you will get a real-time answers, real-time response, which can be observed, and you can decide the thing. Now, we can also go to some cases while teaching, use fate stimulation. This fate stimulation is a very good one. It's a free version, and you just need to log it in. Now, previously I was thinking that it's not having much about biology, but later after logging in, I have seen there are many things which are related to biology, and I'm just going through one of them, which are related to biology for class 11th and 12th. Means for PGT biologists, the teachers who are over here. Now, just try to see this one. May not be exactly like that of CBSE, but gene expression can be explained in this in a nice way. So, how it can be explained? Here are some legends given, like RNA polymerase. Some terms may not match, like that of positive transcription factor, okay, and negative transcription factor. Let it be negative transcription factors are inhibitors, and there are some factors that initiators or promoters. Now, you need to suppose if you take RNA polymerase, and this is a gene 1, a transcriptional region, and if you put it over here, it will not bind. It will just say move from the place. Now, why it is not binding? Because it needs a positive transcriptional factor which should bind over here. And now if you try to put it over here, it will bind and it will do its work, and mRNA will be formed. Now, this mRNA, it needs a ribosome. And it produces a protein. Now, this mRNA, if it is not required, can be destroyed by some other enzymes. What happens if this the positive will bind by here? It shows that is not allowing the process to go on. Now, this is the way how a student can play, and with playful way can get the idea that how transcription occurs, what is the required for transcription. Just so this is into and one more important thing which I have seen over here is that
I think sir has left, sir. due to poor network connection.
Is there something wrong?
Sir has left the meeting. I think poor connection due to. Sir has left the meeting. Yeah, sir is not there.
Okay, we should wait for some more time.
Sir. Yes, sir.
A very interesting session was going on. This Mentimeter is very good, and we can use it in our this webinar also to know the response of the teachers on different questions. And this the fact Colorado that he was using just now to show the interactive simulation is also very interesting. I remember that for the first time when I had learnt the use of computer in biology. It's long back, I think it is in, if I remember, it's 95. It is in 95 when first of all, I have experimented in computer to show this.
I'm extremely sorry,
sir.
sir. I don't know how I have been out of the meeting. It was
please,
some
we were
glitch.
waiting for you.
Yes, sir. I'm just once again coming back to the yes. So, sir, was it visible the fit simulation? This one about
Yes, sir. This is visible.
transcription. Now here I would like to just move to this part. That suppose this is a graph which you're seeing over here. My screen is visible, I think so. A graph is going up. Now, if I increase the concentration of the factors, positive factors high, and make this mRNA destroyer factor low, what will happen to the graph? If I increase the cell from 1 to many, what will happen to the graph? And about the protein degradation, if it is slow, what will happen to the graph? These things can be done, and a student can understand the role of different factors over here. And you know, nowadays, competency-based questions are asked. And the thing the student can develop certain competencies that if the enzyme increases, that this will happen. If the destroyers increases, this will happen. If cells increases in number, then what will happen? So this thing can be seen in the graph by increasing, decreasing the various factors like cell concentration of positive factors, mRNA destroyers, degraders, etc. So like this type of things which are already been made and it is free to use can be used sometimes in class in order to make your class much more interesting. So this is one. And from freight, we come towards another aspect, this one also. We call it as X-Mind. This is basically a platform for making concept maps or mind maps. So once a chapter is completed, we try that the chapter should be remembered easily in a simplest form. And we try to make certain maps called as mind map or concept map. In order to make mind map and concept map and to look it in an easy and a scientific way so that student can remember it well, we can use this app. You can give small project to the students also that they can also make some concept map and you can modify them in this holiday homework. Let them make with one chapter or one part. And this can also help students to learn better, understand better, remember better, and in a short way. Coming towards the next part, this is another Canva for education. You can engage students, inspire students by using Canva. You can make beautiful presentations and that can be used. I'm not talking about how you're going to make it, but I'm just today talking about the various platforms that can be used and you can incorporate them in your studies. In some cases, I'll be showing you some examples. Coming towards HHMI, okay, biointeractive. You see, this one is another. I have chosen one topic in this from this site. That is photosynthesis. Now over here, the topic they have over here with the resource folder, student worksheet. You need not have to prepare. And there are some animations you can show. Now, let's see a worksheet over here. Now, this is the worksheet. They have given the procedure and some questions, and you have to just answer them. Diagram level it. Now, this type of things, if you need to prepare, will take a lot of time, but these are ready-made prepared, and you can just go through, get it free, download it, use it in your class, save time, but make studies much more interesting. The level over here is not lower level because you are seeing about photosystem 1 and photosystem 2, obviously parallel with that of class 11th. And certain what happens in PS2, what happens in PS1, what is cytochrome complex, all these type of things are well prepared in this, and you have a good collections of all this in this particular site. Now, coming back to the animation part or interactive, start interactive. This is an interactive session you can have with this site. Obviously, the only drawback is that you have to have internet throughout while using all ICTs. Otherwise, it is one of the best way how we can engage the students in the class. This again depends upon the speed. At 99, it had been stopped over here. I don't know. Yes, let's start.
Almost all life on Earth is solar powered. Plants.
Let's shift and see some others. This is actually happening due to some network issues. But the thing is, this is really good. You can see each and every part of it. Actually, I'm taking this webinar from a very secure place where I cannot use their internet system and I have to use my mobile internet only. So anyway, this is one part where some interactive part is also shown. And anyway, let's move for the other. We can understand the thing, what is going on over here. So you can get different topics in this. You can search for classroom resources, teaching tools, and professional development tools from this. You can note it down. HHMI Bio Interactive is, of course, a wonderful site where you can create your own account and you can just use these resources in your different classes. Now, coming back to the PowerPoint. Concept maps, this one, Biology-Gautam. Nearpod is another important app where you can also use interactive lessons. I don't know how many of you have used this Nearpod, but this is one important one where you can use it for making your class much more engaging. So let's see Nearpod lesson over here, some of which I have gone through. Let's stop photosynthesis. Let's stop gene expression. You need to sign in. Obviously, in any case, you have to sign in with one of your account. Either you can create your own lesson or you will have a number of lessons over here. Okay. You can just select which one is more suitable for you and you can use it. Say, again, if I see photosynthesis over here and say live participation. There is a code for joining. So others can join by this code. You can share the link or email or through Google Classroom. Or in WhatsApp also, you can share the link. Once you share the link, you can go to the class and just see the material, teacher prepared material, okay? And how to use it, they're showing it. What happens during photosynthesis. And how the class had been divided, engaging and exploring for 5 minutes, explain and elaborate 30 minutes, evaluate 5 minutes. So let's explore. Here are certain initial questions which are given about how much do you know, how much do you already know about photosynthesis? I don't know anything, suppose you say, and you just submit it. Okay, and it will just decide. So anyway, it is nothing like that. It is how much a student say, no one is going to write, I know a lot. I know a little or something like that. And the feedback will be going. The next thing, this is actually engaging the class. But the important thing which comes over here is some videos and some AR is also incorporated. Okay, AR or this you know that augmented reality. That means if you are trying to see a forest or any other place, the video is incorporated in such a way that you can go 360-degree and see the thing over there. So anyway, this also you can use Nearpod and see any lesson, and I think that will enter VR mode. So a typical rainforest, and you can go to a typical rainforest and show that how the rainforest is, and this entire forest is providing food by photosynthesis, and these are the people who are watching in a boat and doing some research work. So this VR and other things all are already incorporated in the lesson. You have only to use them in a proper way in order to make the students understand. Coming towards another important part, biodigital biodigitals. This is basically on anatomy and basically for medical students, but also we can use this one in our teaching to some extent, not in full-fledged. So you can move towards this. I have just selected few parts of it and have kept it. Which requires again email, and you have to just go it in. You can see anatomy and other. So you get all models of anatomy and physiology, section of body, blood capillaries, heart, and etc., brain. These all things, if you can go and have a tour of it, and you can make this teaching, learning much more interesting over here. Much of the part of this library is free and you can explore it. You can see the joints and other things. Even there are some quizzes associated with anatomy and other systems. So this is also a very good platform in ICT using for biology teachers. We can also have some other platforms like what we call as Padlet. This is basically used in flipped classroom like that of when you're giving some work to the students when they're not in class. You have taught something in class and just ask them to give their opinion or view on some questions. Now, one of this is Padlet, which I have prepared earlier. I'm just sharing this one with you all. Now, it is actually a digital interactive board, you can say, or a display board where you can not only put photographs or written matters, you can also add videos. So here, this was the first thing which I have did. What is amniocentesis? How it is done? Is it of any help, betterment for the mankind? What is your view? Go through the attached article, read it, think of it, and comment. And I've given this video, attach the video over here.
Dr. Sahana KP. I'm a consultant obstetrician.
Yes. There is no sound, I think.
So I'm not going very deep in it in order to just only to show you. I'm just going back again. So this video was added and the question was given, and I can show you the feedback, your feedback. So this is the feedback of one student by his name Aryan Kumar, class 12, Bandal. Then another student, Nirjara Kumari, class roll number I've been given. She's of class 12. So after that, I can give some feedback. Excellent, carry on. So you can, other students can give some information. So this is a part. So Anurita Pratap Singh, another student who have given feedback. This process can go on. The student who does not feel good in speaking, hesitate in speaking or giving an answer may also come out by the answer in writing over here. Similarly, you can give some problem like that of this DNA fingerprinting. In the paternity cases, F1 and M1 are one couple, F2 and M2 another couple, child is disputed. Who is the real parent of it? So 85. So like that, different information which you feel a student should go through can have to think for out of the box and make them much more interest in your particular subject. You can make a display board like this, which has no limit. Means no limit means its extension can go beyond your imagination, and student can put information over here, and you can give your own comment. So this is what a Padlet looks like. You can use this one also in your teaching. You can give some work in this holiday. Student can use this Padlet and they can give their responses. And Quizzy is another interactive session. This one, everyone needs a mobile phone that they can answer it immediately. Many of you have did it. This is a question answer series with MCQ type of questions where you can prepare questions or you can give some information. Now AI is integrated with it. You can give certain information of topic and standard and ask to make 10 or 15 questions. They will prepare the questions, give it to you, and you can just use it for your class test. Immediately you will get the response, who is doing better, who is doing fast, who is answering correctly, who is not answering. Everything, every calculation will be done with this quiz. Finally, I would like to end up, because I think I am going beyond the time. I would like to end up with one QR. You can just, you can just scan this and just think, just see what you get. The same way you have scanned, you just scan it and just see what you are getting through this QR. Then this can be a task which you can also do in your school and engage the students for Eco Club and other development. Just scan it and see it. Can you get anything? Are you getting anything? Anyone?
Yes, sir. I got this QR code is for neem plant.
Yes.
So
Now,
the
this is
science
what we are actually
neem and
leveling the plants in our school campus. Thank you,
Yes,
ma'am.
sir.
We are actually leveling the schools of our plants in our school campus, and we have made QR codes in which you can add photograph of the plant, information about the plant, and at the bottom line, there is also another link, the location, the geotag, where it is found. So this was done by myself only, and we were trying to level the plants, but by the time I have got transferred to Baliganj, and now I am in Kolkata. Okay, so these are the few things which I would like to share.
In fact,
And
in
I would
Delhi,
like
all
to...
eco have got this.
Yes, ma'am. Could you
Sir,
please
I
repeat?
was saying that we have also got the same task. We are also doing this in our school, and in Delhi, all eco clubs have been told to
Yes, yes, yes.
do it.
Yes.
So
So
we
this is
are
a,
also in the same. It's a nice
yeah,
initiative.
yeah. So students can also get engaged. They will know the name of the plants and other things with this way. And they should also use the technology, how technology helps in knowing it. No, I have only added a photograph of it because sometimes what happens, a plants may not have leaves or may not have flowers. Flower is the main part for identification. So if a plant is not having flower, that time it is difficult to identify. So at least the student, after getting a scanned one, can find that what type of flower the plants actually produce or fruit the plant produce. So that was my aim. So I have added a photograph. Its economic importance, its brief history of it, and the geolocation, geotag of it. This was the thing. So this was what I would like to share with you all. I'm very much sorry because of internet glitches which I have faced in between. So, ma'am, I should.
Thank you so much, really. And your session really explored us. We can harness the
Thank you,
power
ma'am.
of technology, really, to make the to enhance understanding of the students for this subject. May I now invite Anjali Chipper, ma'am. Anjali, ma'am, are you there to deliver a vote of thanks? It was really a very
Yes,
informative session. Yes.
yes, ma'am. It's a very nice session. Good afternoon, everyone. And it is my privilege and honor to extend a heartfelt vote of thanks to Mr. Gautam Biswas. His lecture was enlightening and thought-provoking on the use of information and communication technology for teaching biology. Sir, your insights and the sites that you have given, the tools that you have told us, they revolutionize the teaching and learning of biology and have broadened our understanding and inspired us to explore innovative ways to make the concepts in biology more engaging and accessible. In fact, with your ability to, make it seem easy, I think all of us will be inspired to use these in teaching, and I would really like to thank you for this. I would
Thank
like
you, ma'am. Thank you very much.
for good to us and such thought, such topics which are in so plus
I think intermittent connection, sir. I think.
Yeah. Yes.
So
कोई बात नहीं.
Sir, please stop sharing your PPT. Vishwas,
Yes.
sir.
Yes. Yes.
Yes, sir. Please. Thank you.
Thank you, Vishwas-ji. It was a power-packed session.
Yes.
Thank you, sir.
sorry,
we
sir,
have
I got disconnected
thoroughly
in
enjoyed.
between.
You want to say something?
No, sir,
No,
thank
sir.
you. I was just saying my thanks to Suman ma'am.
Please,
And I
please
hope
carry on.
all of us,
Carry on.
all us teachers who have been listening very carefully to Gautam sir's lecture, I think we will be using his techniques that he has highlighted today, the tools that he has highlighted in our future classrooms. Thank you, sir.
Gratitude to all
Thank you,
the,
ma'am.
And all the teachers. And all the teachers.
gratitude to Anil sir and
Thank
all
you.
the
Thank you.
speakers, actually. Thank you, sir. Now,
Thank you.